A group of researchers from Monash University have published a paper where they claim that an antiparasitic drug called Ivermectin can be used to stop COVID-19 replication. Ivermectin is approved by the FDA for treating a number of parasitic infections, and the drug has an established safety profile. It can be taken by mouth or applied to the skin for external infestations. Studies have suggested that ivermectin may also be effective in vitro against a broad range of viruses, including HIV, dengue, influenza, and Zika virus. The same group of researchers have already identified ivermectin as an antiviral agent for DNA and RNA viruses. In this paper, they have demonstrated that a single dosage of ivermectin leads to a 5,000-fold reduction of the virus after 48 hours. Now, let us look at how the experiment was done to better conclude the results. The experiment was conducted using a cell line called Vero cells. The Vero lineage is isolated from kidney epithelial cells extracted from African green monkeys. The cell line is widely used to develop drugs and vaccines for over 30 years now. The polio and rabies virus vaccines have been developed using this cell line. The cells were first grown for 24 hours to form a monolayer. They were then infected with the virus for 2 hours before treatment with ivermectin. The results were impressive, and we see a huge reduction in the amount of viral RNA after ivermectin was administered. However, all the experiments have been conducted on a single layer of cells, which is very different from a real organism. They have used a concentration of 5 micromolar ivermectin in 1 ml media on a layer of cells that is only 3.5 cm square. In contrast, the human respiratory tract has a surface area of almost 70 meters square. 5 micromolar of ivermectin in 1 ml media is the same as giving 150 times the recommended dosage currently approved by the FDA. This is like consuming about 150 tablets of a medicine when the recommended dose is only 1 tablet. Also, there is not enough evidence to comment on the safety profile of the drug during pregnancy. We should keep in mind that all of these experiments are done on a plastic plate and they do not correlate completely with the human model. Ivermectin was the focus of a recent phase 3 clinical trial in dengue patients in Thailand, in which a single daily dose was found to be safe but did not produce any clinical benefit. We don't even know if ivermectin will be able to reduce the viral load of SARS-CoV-2 in a mouse model or even a chimpanzee model. It shall then proceed to clinical trials in humans if found suitable. To test the safety and effect of ivermectin on the viral load in humans, it will take at least 1 to 7 years. It is very essential for us to understand that this study in no way tells us that ivermectin cures COVID-19 in humans. This is an initial study that demonstrates that ivermectin may be used as a potential candidate for future research on the COVID-19 cure. We should not take the conclusion of the paper to be applicable to humans. This drug has only been effective to reduce the viral load from a single layer of cells. Rather, it should be perceived as a stepping stone for further research. Self-prescription of drugs can be dangerous and should always be taken under the advice of a licensed doctor.